All right. Lastly, today I'd like to talk about epimorphic limb regeneration of in newt. So in this picture here, you see how you see two amputation of limb in newt, a distal amputation and a proximal amputation, and you see that after a number of days, the animal is able to regenerate the missing tissue, and of course, in both cases, it regenerates only the missing tissue, being the distal portion or the more proximal portion. So how the newt does that? So there are a lot of things that we do not know in terms of how newt does this. It is obviously a very important field of research. These newt are vertebrate, so they're very similar to us, and if we understand how this animal control their regeneration, there is at least the possibility that we could control also regeneration in humans or other animals indeed. So after amputation, after the limb amputation in newt or salamander, the salamander or the newt form what is called a regeneration blastema. This regeneration blastema is formed by cells that are de-differentiated from adult cells. So here we have an example of epimorphic regeneration. So what is this regeneration blastema? If you want, think about the regeneration blastema as a limb progress zone. If you remember, when we talk about limb development, we mentioned the formation of the progress zone. And the progress zone was a group of cells that was dividing very quickly, so a proliferation region. The regeneration blastema is similar to that. It's an aggregate of undifferentiated cells. So these are undifferentiated cells that derive from adult cells through a process of de-differentiation. These cells, after the de-differentiation, will proliferate and re-differentiate in adult type to repair the damage and produce the new tissue that is missing. So we find in the regeneration blastema all kinds of cells. In fact, the regeneration blastema originate from all types of cells that are near the wound when we have amputation of the limb in newt. So we have bone cells, dermis, muscle, cartilage cells. All of these, they differentiate and form the regeneration blastema. So they differentiate, and after a cell division, they redifferentiate in adult type. So what are the different, what's happening when we have amputation in newt or salamander? So the first thing that happens is that after amputation, we have the formation of a plasma clot. This is important, of course, to stop blood loss. So the animal obviously cannot lose too much blood, otherwise it dies. So the first thing is the formation of a plasma clot. Then we have migration of the surrounding epidermis above and to cover the wound. So we have the formation of what is called a wound epidermis. The matrix that usually encapsulates all the cells is degraded, so the cell can divide, but also they differentiate and move around. The tissue-specific genes are downregulated. Of course, muscle cells produce different sort of genes than nerve cells or the dermal cells. So the tissue-specific genes, the genes that identify muscle cells, that identify dermal cells, and so on, are shut down. And on the other hand, the genes that are associated with the limb progress zones are activated. I was telling you that the regeneration blastema resemble kind of the progress zone of during limb development. And in fact, the genes that are associated with the progress zone are also activated here in the regeneration blastema. The surface epidermis also become what is called the apical epidermal cap. And the apical epidermal cap is a signaling center. very similar to the apical ectodermal ridge. So this apical ectodermal cap, and you can see here in green, function more or less in a similar way to the apical ectodermal ridge. Is a signaling center that provides information to the regenerating blastema, which you can see here in red, and instructed to continue proliferating. Very similarly to what the apical ectodermal ridge was doing with the progressome. However, the blastema is not a group of a completely differentiated cells. It's not exactly like, if you want, the progressome. This is because the cells that they differentiate and then proliferate and differentiate again as adult cells retain the memory of what they were. So the master cells, they differentiate, proliferate, and then differentiate again in other master cells. So they don't completely change what they were doing before. They remember what they were doing before, and then they de-differentiate in the same kind of cell. So if you want, the blastema is an heterogeneous assortment of restricted progenitor cells. This is because the progenitor cells will not become whatever, but they will remember what they were before. This should be not surprising to you now, but the apical ectodermal cap stimulate the proliferation of the blastema via FGF8. Why I say that it should not be a surprise to you? It should not be a surprise to you because the apical ectodermal ridge was stimulating the reproduction of the progressome, the reproduction of the cells in the progressome via the same signal. However, one of the main differences here is that this stimulation can only happen if nerves are involved, nerves are present. If we remove the nerves from the damage, then the blastema doesn't even form, and so the limb doesn't grow back. What are other signals that we find associated with the regeneration of the wound? Retinoic and FGF signaling, in the same way that we found in the development of the limb. Wnt, BMP signal and NOT signals. Wnt and BMP, we also remember them from the development of the limb lesson. So they're all signals that we are familiar with, or we should be familiar with, that we already found when we talk about the development of the limb. However, how these signals are associated with the regeneration of the new limb is not very clear. It's not very clear because they are so similar to the signals associated with the limb development. Some of the things that we know are important to underline here is that the nerves are necessary for the regeneration of the limb, as I mentioned to you. However, epidermis signals are also important. Look at this experiment here. In this experiment, a nerve was redirected to a wound, here in gray, of a limb, and a graft of epidermis was crafted near the wound. So here is not an amputation; it's like a cut in the limb. We redirect the nerve to the cut of the limb, and then we craft there a piece of epidermis. This is enough to actually induce the formation of an extra limb. So an extra limb is formed if we have a wound. So we have wound information, wound signaling, we have the nerves information because we crafted there, and we have epidermis information that function as an AEC. But what is the signal that come from the nerves that are so important for the regeneration of a new limb? Well, nobody really knows for sure at the moment, so this is an open area of scientific research. One possible candidate is called the newt anterior gradient protein, or NAG. And to demonstrate how NAG may be one of the signals, if not the signal, that is important for the limb regeneration, and it is associated with the nerves, we have this experiment representing this picture here. This poor animal, this newt, have been used in this experiment, and both limbs have been amputated, but in one limb, in one amputation, the nerves have been removed, so the animal has been de-innervated in that region. And if you observe the regeneration of the limb, what we see is that the limb, the wound with the nerves will regenerate a limb normally, while the other one will not regenerate a limb. So once again, what this is suggesting is that the nerves are important for the formation of the new limb. However, if we treat the wound artificially with NAG, so we add NAG artificially to that region, even if there is no innervation, a new limb is developed. Of course, that new limb will not be functional because we will lack nerves because we remove them, but this demonstrates how this signal, the NAG, may be important for the formation of the new limb and may be associated with the nerves. Okay, so I hope that when you revise and you finish revising this lesson, you will be able to explain the different regeneration mechanisms. You will be able to compare the regeneration mechanism between different animals, and you'll be able to discuss regeneration of the limb in vertebrate, and how the regeneration of the limb in vertebrate is actually associated to the limb development of vertebrate.